So good evening, everyone. We will just wait for 2 more minutes and then we will start with the class. So very good evening to one and all present here. Welcome to the class on Introduction to Complex Biological Systems. I am Srijan Sen and today I will be taking the week 6 live class session which will deal with another interesting topic. Okay, so I'm sharing my slides with you all. So I guess my screen is visible. Okay. If that is so, yes. If that is so, okay. So let us start then. So today's session is mostly on cells, then tissues and organs as a complex architecture of living systems. So we will connect early embryonic development, cell fate decisions, uh, tissue architecture, growth control, then cell-cell or extracellular matrix interactions, and finally the nervous system and neural signaling. So by the end of this class, you should be able to explain how single cell zygote gives rise to diverse cell types and organs. Then you can describe the key mechanisms, the differential gene expressions, asymmetric division, apoptosis, and cell adhesion. You would be able to relate these mechanism to specific examples from the Drosophila, Xenopus to mammalian tissues and the brain. So, keeping these questions in mind, we will proceed with the session. So, let's start from the cycle of life of a multicellular organism. So, at one end, we have specialized germ cells, that's the sperm and ovum, that are produced by meiosis, which we learned in the last class. So meiosis halves the chromosome number, generating haploid gametes to maintain the genome stability across the different generations. When sperm and eggs fuse during fertilization, we re-establish the diploid state and form the zygote, which is a single totipotent cell. Now, this zygote undergoes repeated mitotic divisions to form a compact ball of cells, which we call as the morula. Continued mitosis plus morphogenesis, that is basically the change in shape and position, as well as differentiation, gradually gives rise to an embryo and ultimately an adult body with many tissues and organs. So, the key processes that we need to keep in mind is that meiosis is important for the gamete formation, fertilization to form the zygote, and mitosis plus morphogenesis and differentiation to build the multicellular organisms. So this will help us to actually answer like which processes are involved in increasing the cell number and which in generating genetic diversity, as well as at which stage would you first be able to see the tissue layers forming. Now, the next question that we try to answer is that can someone give one ectoderm derived structure, one mesoderm, and one endoderm? So, this is the next question that we will basically try to answer. So, early in development, most animals form three germ layers, that's the ectoderm, mesoderm, and the endoderm. Now, the ectoderm is the precursor of the epidermis and the nervous system. Endoderm gives rise to the gut and the appendages like the lung and the liver, and the mesoderm produces the muscles, connective tissue. So, the connective tissue and much of the blood and the internal organs. So this same basic body plan is highly conserved across the animals from the invertebrates to the vertebrates. That is why experiments in model organisms can illuminate mechanisms that's relevant to the humans. So these are some of the developmental defects which arise from the misexpression of genes like the eyeless protein called the PAX6, which controls the eye development in Drosophila, and when misexpressed during the development can cause an eye to form in an abdominal site, which is such as the wing or a leg. So you see that the wild type has a normal position of the eye, whereas misexpression of ileus or PAX6 can lead to the eye formation in the wing region. So see, these are some of the uh, difficulties or the developmental defects which rise from the misexpression of genes. So this is basically the conserved mechanism which establishes the basic animal body plan, where there is a dorsal and the ventral side, you have the posterior, anterior, and the right and left. And then when we are now that you understand about the epidermis, endoderm, so you can easily tell that about the positioning of each of these. So developmental potential becomes of cells becomes progressively restricted. So initially, early blastomeres are highly potent. That is, they can give rise to most or all types of cells. As the development proceeds, developmental potential becomes progressively restricted, and cells move from totipotent to pluripotent to multipotent and finally to a fully differentiated state such as the pancreatic islet cells. Now, so basically we can understand that with the gut lineage, a precursor of the gut and the appendages first leads to pancreatic progenitor, which when further specializes into islet cells that secretes the hormones like the insulin. Now at each step, you will see that the functionality becomes more and more sophisticated. Okay, so differential gene expression is largely responsible for the differences between the different cell types. Now, what that actually makes a neuron different from a hepatocyte or a muscle cell? So, differential gene expression is largely responsible for the differences between the cell types. Like nearly all somatic cells, so nearly all the somatic cells carry the same genome, but different sets of genes are turned on and off. Now, cells basically integrate combinatorial signaling, that's multiple extracellular signals into different combinations, and use cell memory to maintain stable gene expression patterns over the time. Now, this combinatorial control allows a relatively small number of signaling pathways to generate a huge diversity of cell identities and spatial patterns. So, if two cells receive the same growth factor but respond differently, what might be different between them can actually be answered through this. Now, let us get into the symmetric and the symmetric division. So, cell diversity can arise via asymmetric cell division. So, in this, the specific proteins or RNAs are unequally distributed between the two daughter cells. So, they inherit different starting conditions and adopt different fates. So, in contrast, you may have a symmetric division, which is followed by extrinsic signals acting only on one daughter, such as secreted factors, or you can say the contact of neighboring cells that then specify the distinct identities. So, both intrinsic asymmetry and the extrinsic cues are used extensively in development and stem cell biology. So from this you can think of like one advantage of symmetric division for maintaining a stem cell pool. Now different animals have used different mechanisms to establish primary axis such as the anterior and the posterior or the dorsal ventral. So in the frog egg, that's the xenopus, molecules are asymmetrically distributed even before fertilization. So defining an animal vegetal axis, fertilization then induces further rearrangements that define dorsal ventral asymmetry. Now, in Drosophila, what you see is basically the egg polarity genes encode the macromolecules deposited by the mother into the egg. So, one key example is the bicoid mRNA. Okay, so which is localized at the anterior pole during oogenesis. After fertilization, local translation and diffusion create a bicoid protein gradient that patterns into the anterior and the posterior axis. Now, another important thing is understanding the growth and the transplantation experiments. So, organ and body size are defined by three main variables, that is, the size of the individual cells, the number of cells, the amount of extracellular material per cell. So, different species and different organs within a species can vary dramatically in these parameters. Now, classic transplantation experiments gives insight into how the organ growth is regulated. For example, when we think of a fetal thymus glands are transplanted into a developing mouse, each thymus grows into its characteristic adult size, regardless of how many are transplanted, suggesting like local organ intrinsic mechanisms which regulate the thymus growth. Now, on the other hand, when multiple fetal spleens are transplanted, each remains smaller than normal, and together they reach about the total size of about one spleen, indicating like a systemic feedback mechanism that senses the total number of spleen tissues into the body. So, uh, so thymus growth is primarily locally regulated, while spleen growth is like a system level feedback control. Now, this principle, which actually fights between the local versus the systemic growth control, is very important when we have to understand the tissue homeostasis. Okay, now we are going into apoptosis or cell death bit by bit. So, um, as I was speaking about the tissue homeostasis, so organ and tissue homeostasis depends balance between the cell division and the cell death. So division includes like mitosis and in germ cells meiosis. So we can distinguish symmetric and asymmetric divisions and stem cells versus the differentiated cells. Now, on the other hand, we have the cell death, in particular apoptosis, we would be mostly discussing about, also called the programmed cell death, PCD, which is an orderly cellular self-destruction process. Now, apoptosis is as crucial for survival of multicellular organisms as cell division is, because it removes the excess, damaged, or dangerous cells in a controlled manner. Okay. Now, the lining of the small intestine is continuously renewed through cell proliferation in the crypts, which we can see like over the time. So you can see epithelial cell migration from birth at the bottom of the crypt to loss at the top of the villus. Then you can see the direction of movement. The top you have the non-dividing differentiated cells, which rapidly divides the cells. The cycle time is 12 hours. You see the stem cells at the bottom of for about 24 hours. That's just an understanding of the lining of the small intestine, which continuously renews through cell proliferation. Now we are getting into the most important part, that's apoptosis, which is an orderly cellular cell destruction program that's activated within a cell that leads to rapid cell death without inflammation. It is essential for growth, development, and survival of multicellular animals and is evolutionarily conserved from nematodes and flies to mice and humans. So, the importance of this process was underscored by the Nobel Prize that was awarded in 2002 for the discoveries on genetic regulation of organ development and programmed cell death. So many of the core genes and stages in apoptosis are conserved across the species. So what are the key features of apoptosis, and maybe how are they different from necrosis? So the key features of apoptosis includes cells shrink and they condense, the cytoskeleton collapses, the nucleus condenses, and the chromatin DNA is fragmented. The plasma membrane builds, but it remains intact, and the cell is ultimately packed into apoptotic bodies that are phagocytosed. Now, in necrosis, what happens is that by contrast, the cells die accidentally in response to acute injury or infection. The membrane ruptures, the contents spill out, and this often triggers inflammation. So, apoptosis is a clean death, whereas necrosis is a messy and damaging to the tissue environment. So, that's a very key important difference between apoptosis and necrosis. So next thing that we want to try understanding is basically apoptotic cells are biochemically recognizable. So they have a chromosomal DNA that gets fragmented. So phosphatidylserine, which is a negatively charged phospholipid, is mostly present in the inner leaflet of a lipid bilayer of plasma membrane, which flips to the outer side in apoptotic cells. Now, this phosphatidylserine now acts as a biochemical marker for the apoptotic cells, and any antibodies against these markers will help you to understand whether the cell has undergone apoptosis or not. So, the apoptotic cells lose the characteristic features of a normal mitochondria. So apoptosis. So mitochondria lose their normal membrane potential, and proteins like the cytochrome C, normally in the intermembrane space, are released into the cytosol where they activate downstream cascades. So the functionalities or the necessity of PCD or apoptosis is that it leads to the elimination of unwanted cells during the organ formation. For example, during digit formation in the mouse paw, apoptosis removes the interdigital tissue, sculpting separate the fingers and the toes. Then the removal of the transient structures in amphibian metamorphosis. So apoptosis removes the tadpole tail as it transforms into a frog. So therefore, it's very important. Then it also puts a quality control on the cells with severe DNA damage that could lead to cancer, undergo apoptosis rather than the risk malignant transformation. Also, in the development of nervous system for matching the cell numbers, apoptosis adjusts the number of neurons to match the number of target cells, ensuring proper connectivity. Now, in adult tissue, it maintains homeostasis in the non-growing tissues. So, apoptosis and cell division are tightly regulated to maintain a stable cell number. So that is why apoptosis is not always a negative effect. It's also a necessity. It's also necessary for most of the cells to maintain proper healthy cells and avoid any kinds of disease condition. Okay. Now we are getting into a new part. But before that, if you have any questions, I can address it, because now we will be entering into the cell adhesion portion. Okay, so no questions? Great. So we will go into the cell matrix adhesions. So um, so the adhesion properties of epithelial and connective tissue. So they have distinct adhesion properties and patterns of cell matrix junctions. So we will focus on the anchoring junctions such as the adherence junctions and the desmosomes, the tight junctions and the gap junctions. So these junctions distribute mechanical stress, create barriers and allow communication between the cells. So yeah, so basically what you see over here is mostly the information about the junctions, where the tight junction seals the gaps between the epithelial cells. You have the adherence junctions that are mostly important for connecting the actin filament bundle in one cell with that to the next cell. You have the desmosomes that connects intermediate filaments in one cell to those of the next cell, the gap junctions which allows the passage of small water soluble molecules from cell to cell. You have actin linked cell matrix junction that anchors the actin filaments in cell to the extracellular matrix, and the hemidesmosomes, which anchors the intermediate filaments in a cell to an extracellular matrix. So mostly the actin-linked cell-cell junction and the hemidesmosomes are among the cell matrix anchoring junctions. You have the channel-forming junction by the gap junctions. The cell-cell anchoring junctions are mainly by the adherents and the desmosomes, and the tight junctions are also quite relevant. So the mechanistic basis of anchoring junctions that support the cell-cell adhesion is quite understood over here. So we had discussed about all of the five ones that have been specified. Okay, now we will get into something about the catenanes and the adherence junctions. Okay, so catenanes are functional only in the presence of calcium ions, which stabilize their extracellular domains. Now, on the cytoplasmic side, catenanes connect to actin filaments via the catenins, providing a mechanical link between the cell surface and the cytoskeleton. Now, it belongs to a major family of adhesion molecules in cell-cell anchoring junctions, the catenins, which are cell surface glycoproteins as well. Now, in adherence junctions, catenins mediate homophilic adhesion, meaning that catenins on one cell binds to identical catenins on the neighboring cell. So, catenin dependent sorting and tissue organization, where catenin expression patterns drive cell sorting during development. Cells expressing the same type and level of catenin preferably aggregate with through, while those with different catenins segregate into different layers or structures, as we see in the early amphibian embryos. So similarly, dynamic Dynamic cadherin expression guides the construction of the vertebrate nervous system, ensuring that specific neural populations find and adhere to the appropriate partners. Now, this shows how a relatively simple principle, that's homophilic adhesion, can generate complex tissue architecture. Now, catenins links classical catenins to the actin cytoskeleton. So the role of tight junctions is that it seals the space between the epithelial cells near the apical surface and controls the transcellular transport by preventing paracellular leakage. Now they maintain polarity by separating the apical and basolateral membrane domains. On the other hand, the gap junctions are channels that couple cells electrically as well as metabolically, allowing the passage of ions and small molecules. Now, this is critical in synchronized contraction of the cardiac muscles and in coordinated responses in most of the tissues. The other part of it belongs to the selectins. So, in bloodstream, cells need transient adhesions. So, selectins plays a role over here. They are cell surface carbohydrate binding proteins that mediate such short-lived interactions. So, L-selectin is found on the leukocytes, P-selectin on the platelets, and E-selectin on the endothelial cells. So during leukocyte rolling on blood cell walls, sorry. So during leukocyte rolling on blood vessel walls, selectins on endothelial cells binds the carbohydrate on the white blood cells, slowing them down, and then integrins on the WBCs bind the ICAM1 on the endothelial cells to form stronger adhesions. Now, this cascade allows the immune system to exit the bloodstream at sites of the inflammation. So, this is the an idea about the extracellular matrix of the animals. So, cells are also embedded in and interact with the extracellular matrix, which is composed of hyaluronan, collagen, fibronectin, which is produced notably by the fibroblast in the connective tissues. Whereas the integrins are transmembrane heterodimers that links the extracellular matrix to the cytoskeleton. They transmit forces and signals in both the directions, inside out and outside in. So the extracellular matrix composition can regulate cell behavior and the cells can remodel their ECM environment. Now getting to the next or the last part of it, which is basically an understanding of the nervous system or uh, the neurons, the brain organization, which is a highly organized tissue, that's the brain. So, as one of the famous founder of modern neuroscience Nobel Prize winner in 1906, Santiago Ramon Cajal said, the brain is a world consisting of number of unexplored continents and great stretches of unknown territory. So our brain is the most sophisticated computational machine which is able to rewire itself and modify its own components to support new functions. That that is it's a plastic. So the Yeah. So this is the different functions that can be mapped to specific parts of our brain. So the neuron doctrine states that neurons are the structural and functional units of the nervous system and they are electrically excitable cells. Now using Golgi staining and the careful drawings, Cahill and Golgi Golgi revealed the cellular organization of the nervous system through which they earned the Nobel Prize. So the function of a neuron depends on its elongated structure. So the more elongated the neuron is, you will see it has greater functions, and if you have greater um, linkages, branches, then the person is said to be more intelligent than the other one. Okay. So now the different regions of a brain are associated with different specific functions. Like some areas are involved in judgment and foresight and voluntary movement, whereas there are some else regions for intellectual and emotional functions, comprehension of language, coordination, and the primary visual area. So, of course, the reality is more distributed and interdependent than the simple maps, but these associations are robust. So, importantly, the brain function depends on the elongated structure of the neurons, with the dendrites receiving the signals, the axons transmitting the action potentials, and the synaptic terminals communicating with the target cells. Now, the geometry of the neurons underlies how networks are wired and how the information flows. So, we understand about the resting potential. Okay. So, but before that, we also need to understand that our brain operates via stimulus response pathway, where our brain and the peripheral nerves operate in the stimulus response circuits. So, environmental stimuli such as the light, sound, touch, chemicals activate the receptors on sensory neurons, generating the nerve impulses. Now, these impulses travel to the central nervous system where information is processed and decisions are made. So, response signals are then transmitted via motor neurons to effectors such as the muscles, which contract to carry out the appropriate action. Now, how is a nerve impulse generated? That's a very interesting question. So, resting membrane potential, which is the electrical difference, potential difference across the cell membrane. So, neurons have a resting membrane potential like all other electrically excitable cells, like a minus 60 to minus 70 millivolts. So the local or the generator potential is when a neuron is stimulated by light, sound, or the local potentials abilities that's being generated. Then the stimulus activates the specific receptors and it lets open the ion channels, then allowing the sodium ions to enter, partially depolarizing the membrane. Now, if this depolarization reaches the threshold at the axon hillock, an action potential is basically triggered. So, we have different types of receptors, like the different stimulus are detected by the different receptor types. Photoreceptors for light, mechanoreceptors for touch and pressure. We have the thermoreceptors for temperature and the chemoreceptors for chemical signals. Now, all of these convert specific physical and chemical stimuli into changes in membrane potential in the sensory neurons. Now, we have some sodium channel blockers where we have the TTX and the lidocaine. So TTX is basically tetrodotoxin found in pufferfish, which is a potent, essentially irreversible sodium channel blocker that can cause paralysis and death by preventing the action potential generation. Whereas the lidocaine is a weaker, reversible blocker used clinically as a local anesthetic For example, in dental procedures, by temporarily blocking the sodium channels into sensory nerves and preventing the pain signals. Now, synaptic transmission and the neurotransmitters. So, at the end of the axon, electrical signals are converted into chemical signals at the synapses. Now, when an axon potential reaches the presynaptic terminal, it triggers neurotransmitter release it into the synaptic cleft. Now, on the other hand, the neurotransmitter binds to the transmitter gated ion channels on the postsynaptic cell, altering its membrane potential and transmitting the signal. So this fundamental mechanism basically earned the Nobel Prize in 1936 for the discoveries related to chemical transmission of the nerve impulses. So some examples of the neurotransmitters includes acetylcholine, dopamine, you have the serotonin, glutamate, and the GABA. Yeah. So that's all about the lecture. If you have any questions, you can ask me or you can put it up in the chat box. We will start with the question and answers after a short break. In the meantime, if you have any questions, you can ask. Okay, so let us go with the questions then. So the first question says, so you people will answer me in the chat box. Okay. So the first question says choose the odd one out in terms of germinal origin. Yes, so RBC, WBC, and nephron structures are mesodermal, whereas neurons are primarily ectoderm derived. So neuron is the odd one out. Okay, let's go to question 2. So go through the question and see what it tells. So it says, suppose you are working in a research laboratory about the development of Drosophila embryo. You have hypothesized that a protein coding gene, that's gene A, can be responsible for wing development in Drosophila. How can you experimentally validate your hypothesis? So choose the correct option: mutating the gene and checking the phenotype, misexpressing the gene in some ectopic site and checking for the phenotype, treating the embryo with translational inhibitor antibiotic like neomycin to stop the expression and check the phenotype, treating the embryo with transcriptional inhibitor molecule like an actomycin D to stop the expression of gene A and checking for the phenotype. B, that is 2 and 4. One person says it's D, 1 and 2. So first what you have to do, you have to check like for whether that particular gene is absolutely important or not. So that is done by mutating or knocking out the gene and observing the resulting phenotype. If the wings fail to develop, it supports your hypothesis. The second is the gain of function analysis that you have to do, where ectopically expressing the gene, that is expressing it in a location where it's not normally found, and observing the phenotype. If the wing develops in an unusual location, it still supports that the gene A is important. But option 3 and 4 suggest providing general inhibitors that would stop the translation and transcription of all the genes, not just specifically gene A. Therefore, it's impossible that you could link the effect of gene A. Okay, so option D, that is 1 and 2, are actually correct. Now getting to question number 3 and 4. So it says lymphocytes, monocytes, and neutrophils are different kind of blood cells. All of them are mesodermal in origin. Which of the following processes can be responsible for the formation of their diversity? Different number of genes present in the genome of these cells, combinatorial code of differential gene expression of these cells, different number of chromosomes in these cells, combinatorial code of gene duplication in these cells. So both of you are saying it's B. So basically, the explanation is all somatic cells in an organism, including different types of blood cells, like the same genome or the same number of genes and the chromosomes. So the diversity in the cells types and the functions arise from the differential gene expression, where different sets of genes are activated or suppressed in specific cell types, leading to the production of different proteins and cellular characteristics. So the correct answer is combinatorial code of differential gene expression in these cells. Okay. Question number 4 says, suppose you're working on embryonic development in mouse model. In two different experiment sets containing one precursor of intestinal epithelium cells, two precursor of hepatocytes, you have treated with the same growth factor. Suppose factor A. So although both of them treated with the same growth factor, experimental set 1 develops into intestinal epithelial cells and set 2 develops into hepatocytes. Which of the following options can be the reason for this? Different structure of double helical DNA in two sets of cells, different number of protein coding genes in two sets of cells, activation of different intracellular pathways in the two sets of cells upon treatment with factor A, or the difference in structure of RNA polymerase. Oh yeah, so there is something problem with the numbering. So there are two A's. So consider one as A, B, C, and D. Yeah. So you're saying C, person saying D. So yeah, the same growth factor can elicit different responses in different cells because the cells have different intracellular signaling pathways and the transcription factors already present or active. So these pre-existing differences determine how the signal from factor A is interpreted and which specific genes are activated or repressed, ultimately leading to the different cell fates. Okay, so question number 4. So I was just getting the connection. So question number 4 is the same growth factor can elicit different responses in the different cell types because the cells have different intracellular signaling pathways and the transcription factors that's already present or active. Now, these pre-existing differences determine how the signal from factor A is interpreted and which specific genes are activated or repressed. So, ultimately leading to the different cell fates. So, option C, that is activation of different intercellular pathways, is correct. Okay, now we'll get into some simpler questions. Like during the first few nuclear divisions of Drosophila zygote, almost all the processes are carried out by paternal RNA and proteins, maternal mRNA and proteins, zygotic mRNA and proteins, and all of the above. So, okay, I got one answer. Anyone else? Okay. So, in the early stages of the Drosophila zygote, the development is mostly controlled by the maternal gene products, that's the mRNA and the proteins that are deposited in the egg during oogenesis. The zygotic genome only becomes active later, that is during the mid-blastular transition. Okay. Now, which of the following statements is so for so for question 5, B is the correct answer. Now, the next one, which of the following statements is true for programmed cell death? So you have three options and one all of the above. So which one do you think is correct? Okay, so programmed cell death is a crucial process for normal development, including I was discussing about the formation of digits in the mouth, paw, or by removing the tissues between them. So option A is correct. So another key event is the flipping of the phosphatidylserine in the inner to the outer leaflet of the plasma membrane, acting as a eat me signal for the phagocytosis. Now, third one, the genes involved in apoptosis, such as the CD genes in the C. elegans, are highly conserved across many species using the humans. So, all the four options over here are actually correct. Okay, coming to question number 7, which is cadherins play an important role in bridging adjacent cells through the tight junction, gap junction, adherent junctions, or all of the above. So, question 7. So the correct option is adherence junction. So catenanes are a type of cell adhesion molecule that plays a critical role in cell-to-cell adhesion. Mostly in the adherence junction, they are calcium-dependent proteins that mediate the bridging of one cell to another, forming a bridge between the adjacent cells. So tight junctions primarily function as a seal to prevent the passage of molecules between the cells, while gap junctions forms channels for the communication. So question number 7, option C is correct. Question number 8, that is the bicoid and nanos are two important proteins that dictate the development of Das axis in the Drosophila. Is it the anterior posterior or the dorsal ventral, right left, or animal vegetal? So mostly in the early development of the fruit fly Drosophila, the proteins that's mentioned, the bicoid and the nanos, establish a primary body axis. So its bicoid is concentrated at the anterior head and nanos is concentrated at the posterior. So these concentration gradients dictate where the head, thorax, and abdomen will form, thus establishing the anterior posterior axis. So option A is correct. And the last two questions. Yeah. So which of the following cytoskeletal proteins play a crucial role in the adherence junction? It's very easy. So actins are proteins that form microfilaments, one of the three major components of the cytoskeleton, which is essential for the establishment and maintenance of cell junctions and the cell shape. So adherent junctions have a cytoplasmic phase which is linked to the actin cytoskeleton. So actins is the correct answer. Option C. And the last one, which is which of the following ion level across the neural cell membrane play the fundamental role in generation of action potential? Sodium ion, potassium ion, calcium ion, or both A and B? So it is also quite a direct question. The generation of the action potential in the neurons fundamentally involves the rapid changes in the levels of both sodium and potassium ions across the cell membrane. So the influx of sodium ions causes initial depolarization, where the flux of K plus ions is responsible for repolarization and hyperpolarization. So option D. Okay. So that's all from my part. If you have any questions, any doubts from any of the previous classes regarding any assignments about this class, any questions on this part, you can ask me. Any questions, any doubts, any clarifications? No? Okay then. Uh, Thank you for joining. We will again meet in the next week on Friday at 6 p.m. and then we can discuss another interesting topic. Okay. Thank you for joining. Have a good day.